I mean, this APCC, that's the same APCC that functions during mitosis and that functioned during meiosis 1. Okay, so so there are some more commonalities here beyond just what's happening in the oocyte. Yeah, okay, so let's look at this in a little bit more detail. Here's the metaphase 2A before fertilization. And remember, this is what gets ovulated, arrested in metaphase 1. We have that first polar body. This is the reductional division. Now we get the sperm coming in. Of course, as we saw on the previous slide, this was triggering a whole bunch of things, including the onset of anaphase. Okay, and as I showed you in the video, the live video the other day, if you're interested, I'll post that. Okay, this spindle has to move to get close to the periphery, right? Because we've got to have the polar body going to the exterior. And it's got to be lined up in a way that the mitotic spindle, those daughter cells, would be going in opposite directions, right? You wouldn't have the spindle oriented so that the division is taking place like that. Okay, so that's all tightly controlled. Now, I can't remember if I had, what APCC, I have this on the slide, I think. No, I don't have it. APCC actually activates this enzyme called separase. That's really easy to remember. What separase does is cleave the cohesins. Now, typically we think of cohesins as surrounding the sister chromatids after S phase, but that happens in meiosis surrounding there's 3 proteins in the complex and it literally forms a circle. 2 proteins on the lateral side and 1 like a connecting bridge, okay? That literally forms between those, holding the sister chromatids together in meiosis, but in addition, particularly at the arms, it wraps around the homologs. Okay. So what it does is triggers an enzyme that cleaves cohesin. You don't have to know these terminology. REC8 is that bridge that allows the circles to go around the chromatids. Separase cleaves REC8. That releases. Now, if you might remember this, I think I know it's discussed, I discussed this in my 3200 section. In meiosis 1, separase cleaves the cohesin just along the arms. And there's another protein that protects the cohesin at the sister centromeres. That's why the homologs don't separate in meiosis 1. Here, we don't have, and when we're completing meiosis 1, then there's no cleavage that needs to take place on the arms, but separase does cleave this cohesin at the sister centromeres. Okay. So again, this is all tightly controlled. As far as I know, now there are some meiotic-specific differences. So one of this REC8 is only in meiosis, and there's an equivalent protein, SCC something, don't worry about it, that replaces it in mitosis. Okay, so even though the general idea of cohesins and the general structure of cohesins is the same between mitosis and meiosis, there are some fundamental differences. And that's also kind of fascinating when we think about that. The process of meiosis, especially in the oocytes, there's all kinds of things that are different from mitosis. So it kind of makes sense that there's some proteins that are a little bit different. Okay. Now, the other things, and this is summarizing everything, and this is again from that review article, and really pay attention to, for this I think both on the review article, I added some text to the PDF, and I have it on the class schedule, the parts of it that you should pay attention to. Okay, and then the other parts that you don't need to. You can read if you want. Okay, this is just summarizing everything and showing the chromosomes. But we've already talked about the location and orientation of basically the meiotic spindle at the time of the second polar body formation. But the other thing that I just think is fascinating and has some very potential significance, particularly to aneuploidy in humans, and I'll get back to that in a few seconds. The meiotic spindle in females, as far as I know, is all animals. Female, so the oocytes only, the spermatocytes don't have this. The oocytes lack the centrosome. How can you have a spindle without a centrosome? Well, in some structure, were you going to say something? Yeah. So in some organisms, there's still a microtubule organizing center. But that has to happen. So it's functioning like a centrosome, but there is no centrosome. So one of the remarkable things that has to take place, and I'm not going into detail here, but remember after we have that zygote form, the two pronuclei fuse, there's a couple days it's before this, okay? And so after this stage, this spindle has to switch from having, there still is one spindle here in this oocyte, right? And there's a spindle from the male, okay? Because that has to form a mitotic spindle. It makes that transition from being, on the female side, being acentrosomal to centrosomal. And if you're thinking, well, what's the big deal about that? It's been pretty well studied, or there's a very active area of research with this, both in mouse as models, but in humans. Okay, and as you can imagine, there's severe limitations, and I think you can think of them, to studying oocytes in females, in women. Okay, a lot of things can be studied in mice. Larger numbers, more control, all that. Don't have to worry about ethical issues, all that. But there are some studies being done in humans. Lots of studies done in mice. You have a question?
It's kind of just about like when this is talked about in the paper. And it would also talk about the lower vertebrates and all of that and amphibians and everything. Do we need to know all those details?
No.
Okay.
I'll point out when there's some differences between human and mouse. Only because I'll be giving anyway multiple examples of mouse models. But what I'll do is point out where there's differences between mouse and human. Okay. So that's a good question.
Thank you.
If you're interested, yeah, of course, read them, but you won't be tested on this. Was it on the study guide to know anything? Now, I'm not saying anything about human, but unless I write something about mouse specific on a study guide, you can assume all the questions are about human.
Okay, perfect.
Yeah.
Thank you.
You're welcome. So interestingly enough, there are some differences between human and mouse. Mouse oocytes are still acentrosomal, but I just wanted, in case you're not really fully appreciating it. Now, if I hadn't told you, if I didn't have it labeled, I think it's under here. It's not. This is telling you this is an oocyte, but I think you can imagine, well, there's the first polar body. That's the metaphase spindle. But look at it, it's like a barrel. This, would you be able to identify this as a mitotic spindle? This is practically the textbook case. Actually, I took it from Wikipedia, because it's a nice one. There's where the centrosome is. There is no centrosome there. And now these are not colored fluorescent images, but this is actually, you can get an animation of this. This is actually in humans, and it's just showing you the difference. I mean, that barrel's right here. We're just seeing the spindle fibers. That's the lightly staining stuff here. It's actually fluorescently stable, but this is just a black and white grayscale image. And you don't see the chromosomes, but they would be here. Metaphase 1, metaphase 2. And this was from a 2015 paper, and this lab, Melina Shu, is in Europe, and she's the one that's really doing a lot of this work, both in mouse and human, and it's really fascinating because, again, now, we don't know exactly what the contribution of this lack of the centrosome has to the high extent of aneuploidy in humans, particularly in oocytes, but this is a very good starting point, or very good reason to think about that and consider it, and that's what people are doing. Okay, now we're back to the posted slides. Again, this is from the review article. Now, we've already gone over some of the things happening. These are the same figures. We're still just up to the first mitosis. But I just want to remind you of some other things. Now pay attention to this color coding. This red line is the maternal mRNA levels. And remember, their translation is inhibited at this point, but then they get, they start, as you saw in a previous slide, the maternal mRNA start being translated here. But look at the embryonic expression, the zygotic expression. Now this is actually kind of a low level. It's been hard to define exactly in humans, and it is a little bit different in timing in mouse and human, but so this is just an approximation. But essentially, you don't get the highest level of embryonic expression until the two-cell stage. So up until this point, everything, all those events that are taking place in fertilization and setting up this first mitotic cleavage is almost exclusively dependent on maternal gene expression, what was stored in that oocyte and activated for transcription. Okay. In addition, chromatin remodeling, switch SNP and a related protein. Remember, and we'll see this in another more detail on the next slide, there are massive changes taking place at the level of epigenetics. Anybody remember what switch SNP is? Is that a histone modifier? Does it acetylate or deacetylate DNA? What's switch SNP and what? That's chromatin remodeling. Is that histone modification? Switch SNP is the prototype of, it's the first family member of a pretty large family now. It was originally identified in yeast, but it's been identified, I think, in all eukaryotes. But that moves aside the nucleosomes. Okay. No histone modification, we're not talking about that, although I'm sure it's related. But there's all kinds of things that are taking place at the level of the epigenome and these at this embryonic stage. And we'll see, okay, there's where the translation and decay is activated. Now this, I know there's a lot of detail, but I think you can follow it along. And really, in terms of, you don't have to know, I think I covered up, if you look at the figure in the book, this is telling, I covered up the actual enzymes that are involved in this. If you want to know about them, it's fine if you look it up. You won't be tested on what is the enzyme that demethylates the paternal genome at this point. But I do want you to be aware. Okay, so first of all, the main point is that all of the maternal and paternal genome has to be completely demethylated. Well, not quite. Just pay attention. So we've got the pink and blue. It's different between male and female. What are imprinted genes?
Silencing?
Are they what?
Silencing? Wait.
Well, they are silenced. Remember, there's paternal imprinting and maternal imprinting, and if yes.
It's patterns of methylation when you read it before actually fertilization occurs.
Yes, and they're exactly the same between male and female, right? No, there are some genes. Remember, this is only a pretty small set of genes, but some genes get paternally imprinted. Other genes get maternally imprinted. So that, kind of surprisingly enough, those methylation marks, they're literally called marks, epigenetic marks of DNA methylation, are maintained. They're protected from this demethylation that's taking place on all the other genes. And I'm not quite sure why, but there's what's called active demethylation in the male. That happens first, and that's actually driven. There's an enzyme that actively removes that methyl group. In the female, it's actually called passive demethylation, but basically it's just thinking about diluting the DNA, because now we're getting mitosis. We're getting multiple rounds of S phase, so multiple rounds of mitosis, S phase, and there's no active methylation taking place yet. So this is called passive in the female, because, and actually there's a certain amount of passive demethylation in the paternal also, just because of dilution. The newly synthesized DNA doesn't get methylated until later. Yes?
This might be a dumb question, but like how?
There are no dumb questions that can be asked in this class.
So then, like, I'm a little bit confused on this side. How is the cell able to tell that the paternal genes are like unventilated?
Wow, that is an excellent question. I'm not sure. There are some suggestions. But I'm pretty sure that's not a dumb question at all. It's a perfectly good question, and I'm not sure if there's a conclusive answer yet. First, yeah. They do have different marks. Now, remember this protamine exchange? I mentioned this kind of in passing the other day, but I think you can imagine that the chromatin genome compaction in the sperm is a lot more tightly compacted than in the oocyte. And the reason that can take place, that can't occur if the nucleosomes are consisting of the regular histones. So before, during the later stages of spermatogenesis, I forgot exactly what point, but all the histones are replaced by protamines, which are functionally different. So the first thing I would guess, and this is just pure speculation, there's perhaps something about that chromatin structure that it allows it to be recognized. There is some enzyme. The review article might explain that in more detail. Might have some clues. I don't know. I don't expect it. But that is an excellent question. Anytime I say something that happens specific, one of the next things, that sets in motion a bunch of things, something gets activated and then things. Next thing you should be thinking about, well, what's happens to the before that step? What's activating that first step, right? Okay. So the other thing that we'll talk in more detail about, we go from, look, we've got this mitosis going on, and it literally goes up to the 32 or 64 cell stage, or 8 cell, it's on the next slide. There's some things that happen in this ball of cells going through mitosis, but up until around here, these cells are undifferentiated. They're genetically identical, right? Mitosis. In vitro, in IVF clinics, assisted reproduction clinics, these embryos would be in culture. You can, as we'll see, you can do pre-embryonic genetic diagnosis. You can remove a blastomere. Each of these cells at this stage is called a blastomere. You can remove 1 or 2 or possibly even 3 cells, and the embryo is perfectly fine. Okay. But then there's this decision that's made. See the color coding here? These yellow cells, and they have a different shape. They express different genes and have an entirely different function. This is the first lineage commitment, the first cell fate decision that's made. And that's what we'll talk about in detail, and then other things take place. Now remember, this is all pre-implantation. Okay, now I do want to spend some time talking about aneuploidy. And I'm not going to finish I will finish today's lecture. We will get into embryonic development. I won't finish embryonic development today. It will have to be continued on Tuesday. Okay. I think you all know about aneuploidy. The first thing I want to remind you, especially with humans, we most often are thinking about meiotic aneuploidy that results in non-disjunction like trisomy 21 Down syndrome. But I want you to realize that that's not the only chromosome that can be aneuploid. We now know from many studies that any of the human chromosomes can be in the wrong number. It's just that trisomy 21, because of the genes located on chromosome 21, that trisomy is the only one that allows relatively normal lifespan. Some of them, and we'll see in a few minutes, aneuploidy for some of the chromosomes, depending on which chromosome it is, whether it's monosomy or trisomy, will actually result in embryonic lethality. Okay. So don't be thinking that non-disjunction can only take place with chromosome 21. It's only the one that allows viability. And the other thing is, I don't want you to be thinking it only occurs in meiosis. As we'll see, there's a lot higher incidence of aneuploidy taking place during mitosis in those very early embryos than we knew about before. and it's hugely significant. I've got some other slides. Aneuploidy, not disjunction, aneuploidy is a huge issue with humans. We're terrible as a species with respect to lots of biological properties, but this is one of them. Now, the other thing I want you to be very aware of, because this happens all the time, we'll see some examples of it in embryos, the difference between constitutive aneuploidy and mosaic. So constitutive aneuploidy, most of the time, would be taking place because of non-disjunction in the gametes, or real early, at, even in that first mitotic division or the second one. It's essentially constitutive when, if you look at all the cells, either in the embryo later in development or in the postnatal, all of the cells have the same aneuploidy. There is a large amount of mosaic aneuploidy, where some cells are euploid, remember that's the normal number of chromosomes, and some cells are aneuploid. That almost always has to take place in mitosis. And it can take place really at any stage. It can take place in the blastomeres, we'll see some examples of that. It can even take place postnatally. And yeah, it happens a lot. So constitutive and mosaic, and I'll show you an example later on of probably the most well-known case of mosaic aneuploidy. Okay, so this is the slide. It's a huge problem. This aneuploidy and non-disjunction, whether it's meiotic or mitotic, is the leading cause by far of miscarriage and congenital birth defects. But even the data that we have for this is probably an underestimate, because a lot of times the aneuploidy that occurs really early in development, women is before the women even know they're pregnant. Exactly. Do you know, and I don't know this, at what stage after, days after fertilization, is there enough of that hormone in the blood to be detected as being pregnant? 2 weeks after? Oh, I didn't think it was even that fast.
Yeah.
Okay.
Sometimes it can be 12 days if some women produce a lot of that hormone really fast.
Okay. But there could still be, as we'll see in the timing, there could still be a lot of spontaneous miscarriage and she had no clue of being pregnant. Okay. Yeah, now this is the kind of thing you won't be tested on the details of this, but this is a general thing I do want you to know. This is a huge issue in human genetics and human disorders. Okay? Now, I do want to go through a couple minutes. The karyotyping, that's the standard cytogenetics. It could be done in old fashion. These days, is almost always done in incorporating fluorescence in situ hybridization with fluorescent DNA probes with microscopy on chromosome spreads. I've got a couple examples of that. Single cell whole genome sequencing. People are doing this now with the cleavage stage blastomeres, taking individual cells from a 4-cell embryo, 8-cell embryo, 16-cell embryo, and not even just doing DNA sequencing to look for chromosome abnormalities, but they're doing RNA-seq. So determining the transcriptome of individual cells in a 4-cell, 8-cell, 16-cell embryo. And it's revealing some amazing things. They're not nearly as uniform as we would think. Okay, and that, again, has some significance. I'll show you a couple examples of fish, but I want you to know about ACTH. Okay, so first the standard karyotype. So look at these. There's 4 different individuals. Anybody want to tell me what's wrong? What makes this one aneuploid? First of all, tell me the set. Do you remember the standard cytogenetic nomenclature? What's the first thing you have to indicate? The number. How many chromosomes are here? There's 3 there, right? Yeah. So that's the way you would write that. And this is the trisomy 21 for a male. Same thing. What's happening here? Where is it? Ah. Now, trisomy 13, I showed these examples, trisomy 13 and trisomy 18. Both of these do allow One is called Patau syndrome. I've got a slide. Forget it. Some individuals with these trisomies can be born, they don't survive very long, and they've got pretty profound developmental problems. What about these? XO. On a single sex chromosome. Triple X, these are, as far as I know, I don't think they're viable, or very few of them would be born. Oh, look at this one. XXY. Ooh, XYY. Okay, now fish, not the organism. Fluorescence and see-through hybridization. So these are DNA probes. Now this is for specific chromosomes. These could be large chunks of DNA. These are, you can actually do fish pretty well with oligonucleotides. So 40 nucleotides of DNA. Okay, it just depends on what you have. You can do multiple colors like shown here. Okay, if let's see, did I tell you what's gone here? But if I didn't tell you this is a trisomy A, could you figure it out? Pretty simple. Okay. Now, just so you know, you don't have to have the chromosome spreads. These chromosome spreads would have to be done with cells in culture, and they'd have to be at metaphase. You can also do interphase fish. And for this kind of analysis, just aneuploidy, this is very simple. This would be hard, you can do this to a certain extent with translocations and inversions and stuff like that, but it just kind of depends on the level of detection. Oh, there's the nomenclature for that. Okay, now this is just showing you how PGD, pre-implantation genetic diagnosis, would be done. Obviously, this would be in an assisted reproduction clinic, okay? This would be virtually impossible to do in a human. This pipette, this is under the microscope. This pipette is just applying just a tiny little bit of suction, but it's just to hold that oocyte in place, okay? They would be, that you could be watching, the technician would be watching it, because some of these divisions will be taking place pretty quickly. And now what you see here on the right is, this is an even smaller pipette, and sometimes this is done by real tight control with gas. Sometimes people in the old days, and they still do this regularly with mice, it's not literally a mouth pipette, but there's a little thing that you put in the mouth, and there's a long tube with a very drawn out, very thin pipette that you just kind of draw. Okay. But the point is that you can just very gently, you got to get through there's a structure. Anybody remember that special structure that's around the oocyte? The zona. So you have to get, that zona is kind of tough, so you got to push just hard enough to get through the zona. Then you just remove a couple cells, and then just let the embryo continue on developing, no problem. And then you can very easily now, because there's only a couple cells, you wouldn't want to put these in culture, right? Because if you put them in culture and let them divide, you don't know if the non-destruction would be taking place in the cells in culture, but you can very easily, the experts can do fish, interphase fish on these blastomeres. And again, this is showing 5 different colors. Yes?
Is there any issue with leaving an odd number of blastomeres?
As far as I know, not. It's a good question. And actually, we'll see that you would think that after the first cleavage, right, 2 cells, you would think that both, we'll see this in an animation, you would think that both of those 2 cells would divide at the same time, but actually there's pretty clear evidence that one of those cells actually divides first. So temporarily there are 3 cell embryos. So that's why I'm thinking there's probably no effect of having an odd number. Yeah.
And you talked about interphase fish. What is the benefit for having interphase fish versus other phases?
Well, interphase fish, where you don't get the nice compacted chromosomes like on the standard karyotype analysis, you don't get as much resolution. Okay, but that has to be done with cells. Remember, those are metaphase chromosomes. Those are in cells that have been put into culture. They've probably gone, well, they have to go through multiple rounds of cell division and mitosis, right? You would not be doing that with this. Right. The amazing thing, and we'll see evidence of this, the amazing thing is that even these cleavage stage embryos can have mosaic aneuploidy. Yeah. Yes?
I know humans have like long development time where it would be easy to pull cells out like this, but is there any research indicating that there'd be damage if you did it in processes differentiating?
As far as I know, it's only done in these cleavage stages before the blastocyst.
How do they know?
Oh, well, that's not quite true, because they typically we'll see this in a few minutes, but in the blastocyst, remember I pointed on the previous slide, there's some different colored cells that are in a group at one end, and then the outer cells are different. Those are actually trophoblasts. So at that point it's perfected germ, but they develop into trophoblasts. That is derived from the embryo. They can do, I think it's actually much more common and less much of a risk to do trophoblast biopsies. Yeah, no, good question. I'm not sure exactly when that's done. Okay. Yeah. It would not, I wouldn't think it would be done after implantation. It would have to be before implantation. Okay, so everybody comfortable with microarrays, chips? So in other words, there's hundreds of thousands of spots on these little arrays, these chips shown here. Each spot has a specific piece of DNA. These can be oligonucleotides, or in the older days, they could become fragments of DNA. You can have the whole chip encompass the entire genome, or you could have it, if you were just focused for research purposes, you wanted to have high resolution just in a couple different regions. For this particular technique, this would encompass the whole genome. And you probably talked about transcriptomes, looking at using microarrays for transcriptomes. So remember, you'd have two populations, two different types of cells. You'd purify the RNA from one, label that with one, make cDNA, label that cDNA from that first cell type with one fluorescent color, get cDNA from the other cell type, label that cDNA with a different color. You'd mix those two cDNA samples together and basically let them compete for hybridization to the same spots. So if one has one particular message that's a lot higher level than the other, you'll see that color. If they're equal, you'll see most of the time this is done in red and green. If they're equal, you'd see yellow spots. Or you'd see red, depending on which gene. So you get massive amounts of DNA in that entire transcriptome. This is all analyzed by software. Lots of data manipulation, statistics involved, and normalization and stuff. But now, the same concept is being done with array-based comparative genomic hybridization, ACGH. But instead of taking RNA from two populations, you're taking DNA. There would always be a reference control DNA, and it would have been just it's probably a composite of DNA from lots of different individuals, but the lab that's doing this would have one control, huge amount, milligram quantity, that's a huge amount of that DNA, and always use it as the reference, the control, because what they're going to be doing then would be comparing the patient DNA to that control. Two different colors, let those DNAs compete for the same spots. Now, the data representation is a little bit different. What we'll see on the next slides, this is represented, here we've got blue, and, well, they're showing green and red here, but they're showing blue. This is from the same figure, huh? Anyway, this is just where it's relatively smooth. There's equal hybridization between the control and the patient. What this line represents is the entire genome, the whole thing, because you're doing genomic hybridization. And there's some statistics involved here, because there's going to be a little bit of variation in the signal, Okay, it's got to be kind of smoothed out. But see where this is higher? This means that there's extra DNA in the patient. And what we'll see is that each of these regions are divided into chromosomes, going from the end of one arm to the other arm lined up, just, what's that term? Head to tail, or concatenated. If this is higher, this is lower. Okay, so I've got some examples here. Now these are from blastomere biopsies. This is just one example. This is one embryo. This is in bleeding. This is another embryo. So now see these gray, these slightly darkened regions and the numbers? From one arm of the end of one arm of chromosome 1 to the end of the other arm of chromosome 1. And if you wanted to zoom in on particular regions, you can. You know that this exact alignment all the way, this exactly corresponds to the nucleotide sequence in the human reference genome. So you could, if you wanted to, you could zoom in on this particular region or this particular, whatever region you want. For the purposes of this analysis, they're not caring necessarily about individual regions. Now, yes, you can use the same technique to look for translocations. Well, no, wait a minute. Copy number variants. Yes, you could have small deletions or small duplications that could be detected by this. It would probably require higher resolution. We're just looking for non-disjunction. So this is a blastomere from one embryo. This is a blastomere from another embryo. So what do you see in this top result? An extra, so trisomy chromosome 4. Can you tell me the sex of that embryo? I haven't told you something that is important, but I think you can figure it out, especially, so it's kind of cut off, the X and the Y, there's the Y. Sorry about that, I think, can you see it on the slides, is the Y chromosome written on the, okay. But,
So it's missing the Y chromosome, it has 2 X's.
yeah. So the control DNA is XY. Could you use XX? Well, yeah, I guess you could, but I don't think it would be as accurate. So this is a XX individual, XX cell that has trisomy 4. This is the other thing I wanted you to see about this. It's not just one chromosome. You can have multiple aneuploidy for multiple chromosomes. Now this is from one cell. So there has to be some amplification of DNA in order to do this, and I don't even know the details of that. But we'll see an even more stunning example of the. Is this constitutive or mosaic aneuploidy? This bottom one. We don't know. This is just one cell. Remember, constitutive refers to whether different cells are either aneuploid or euploid. Okay, I think you know these terms already, nullosomy, monosomy, trisomy. These are the chromosome 13 and 18 that can survive. But look at these, there are some chromosomes, and I don't think I have the details of this, but monosomy or nullosomy for some chromosomes will actually allow a little bit more development, nullosomy for others, but basically nullosomy, so that completely lacks a homologous pair of chromosomes. So none of those genes. Remember, each chromosome essentially has different genes. Monosomy, embryonic, trisomy, mostly lethal during embryonic retrieval, with the exception of these 3. Now, you don't have to know the details of trisomy. I just wanted to put this slide up here if you want to read more. And I just want to tell you that just in my lifetime, okay, I'm 63, just in my lifetime, the attitudes of the general public to individuals with Down syndrome has just had a world of difference. When I was growing up, basically people thought there wasn't any hope of an individual with Down syndrome, integrating fully into society. and that's changed a lot. Right. If I ever give examples of some disorder or syndrome like this, and it has a, you have a personal connection to it, just let me know privately if you're uncomfortable talking about something like that in class. If you want to share, that's perfectly fine. Okay, if you have a personal experience, but I don't want to make anybody uncomfortable. Now, these are the constitutive sex chromosome aneuploidy. XO, they're viable. XXY, they're viable. So aneuploidy for autosomes have much more profound problems. Aneuploidy for sex chromosomes is different. Single Y, lethal. Now, the thing that's interesting that you might not know about Turner syndrome, most individuals with Turner syndrome, XO, are actually mosaic. Because constitutive X monosomy is lethal in the embryo. And you can actually have pretty mild forms of XO. You might have very mild forms of Turner syndrome. You might not necessarily know looking at them that they have been diagnosed with Turner syndrome, but that would be because their mosaic aneuploidy is such that they don't have very many aneuploid cells.
You know why they get some of the symptoms that are unrelated to like sexual development? Like obviously rudimentary ovaries and no menstruation makes sense, but like an elbow deformity or like a folded skin.
Yeah, that's a good question. Now this one I can answer, the short stature. They're pretty sure there's good evidence for that. That's a good question. I don't know about all the other aspects. Excellent question. It could be that there's gene dosage for a gene on the X chromosome that's affecting expression or function of an autosomal gene. That would be my first guess. Because I wouldn't think necessarily that any of the X-linked genes would be directly associated with some of these things. Yeah, another good question. The other thing that I think is fascinating, and there's lots of data on this, in the vast majority of cases, it's the maternal X that's present. It's the paternal X that gets lost. Now, would this be this kind of, so is Turner syndrome, especially the living Turner individuals, is this likely to be non-disjunction taking place in meiosis or in mitosis? This is a trick question. Mosaic. Mitosis, yeah. Or because how else would you have cells that have a different karyotype, right? And presumably, now there's a number of things that could affect the cell division of the cells that had the non-disjunction event. So it's not a hard, fast rule, but generally you would think if an individual has more aneuploid cells, a lot more, you would think that had taken them the non-disjunction had taken place earlier, especially if it was like it should be affecting lots of different types of cells. Okay, now I do want you to just think about it, since aneuploidy and non-disjunction is such an important aspect of humans, how would we go about doing research on it? Clearly we need to, we want to know the mechanisms, right? We'd want to know if it's the same mechanism affecting every chromosome. Think about the difference in size of different chromosomes, think about the genes that are present on different chromosomes, think about the heterochromatin status of each chromosome, the position. There's all kinds of things that could be affecting it, but we'd want to know that. And we want to do it with early embryos, right? Because since that's where by far most of the non-disjunction is taking place. But what's the problem with looking at something like any biological process in real early human embryos, pre-implantation human embryos?
Ethical discussions about that.
Ethical. Yeah, exactly. But let's think about the scientific. Yeah. Yes.
Most women don't even know they're pregnant, let alone enough to be able to possibly procreate before
Right, so how could these studies be being done then in the early embryo? What? Yeah, in vitro. And in vitro requires taking those cells out of the mom, taking the sperm from the dad, putting them in cultures. And the scientists do everything they can to try to make the culture conditions as close to possible as what's happening in the woman, but do we know exactly what's happening in the woman? That's very difficult to determine. All they can really determine in vitro is whether the cells are grown, look happy and healthy, and whether they seem to be going through all the steps that they should be taking. We don't know if it's probably not exactly like what's happening in the mom, in the woman, but we know we get viable, they know, that they get viable embryos from that, and viable offspring, so it's working well enough, but we don't know if there could be things in the culture conditions that could be influencing chromosome segregation or the cell cycle or any number of things. So yeah, we can get data, assuming we can get enough ethically and numbers-wise collect enough of them, but who knows if it's valid. Now yeah, we can use mouse models. Mice are very similar to humans in many ways, but we're not mice. So how valid is it? And I've already told you about, even though mouse oocytes and human oocytes are both acentrosomal, what I didn't tell you is that there is, even though both species lack the centrosome in those meiotic spindles of the female, there is a huge difference in how the spindle is organized. We know that much about mouse versus human, so right there's something that could be affecting mouse. So it's a really important problem, but really difficult to study because of the biological process. So just to go through a few things, and again, I could give you more review articles if you want. I mean, early on, even in the 50s and 60s and 70s, even up into the 80s, people were studying these and knowing that it was at least live-born, limited to a couple chromosomes. But look at this. Once they started doing pre-implantation genetic diagnosis, 50% of cleavage stage embryos pre-implantation are aneuploid. 50%. In culture, does that mean 50%? In vivo, we have no idea. The level of mitotic is probably a lot higher than most people would think. The level of mosaicism. I've just got a couple more slides and then we'll take a break if you're getting tired. Okay. And this is very recent data. And this was doing, as I recall, whole genome sequencing of literally tens of thousands of embryos from IVF clinics. Yeah. So you don't have to know the details of it. So, but this is looking at 3-day embryos from IVF clinics. Whole genome sequencing, but the whole genome sequencing was used to identify non-disjunction. So remember the age-related increase in women and increased incidence of trisomy 21, that's very well known. So older women have a much higher risk of having a child with trisomy 21. So a big question would be, does that same trend hold for pre-implantation embryos? And it absolutely does. This is the age, look at the age. Maternal age, young, older. Color coding, maternal, meiotic. Paternal meiotic, no age-related increase. Mitotic, no age-related increase. So these are examples of the mosaicism that is being seen with these blastomeres in IVF clinics. So this is one individual, but look, one blastomere 1, blastomere 2. This is from a 4-cell embryo. This one happens to be identical. Okay, so at least these 2 out of the 4 appear to be constitutive. Look at this one. Chromosomes 2, 7, 16, 19, and the sex chromosomes. This, I can guarantee, would not survive very long, but look, this is interesting because I think, at least for the autosomes, they're reciprocal, I believe. So this one is lost chromosome 2, that's gained chromosome 2, this is lost chromosome 7. So this is kind of interesting. Clearly, you would think then that this took place in mitosis because they're reciprocal events. I didn't actually look at the X and the Y chromosomes. And okay, this is a blow-up, expanded view of that second set of data from the previous slide, just to show you in higher detail. X, Y, this is the Y, O. Okay. Now this is kind of stunning. 8-cell embryo, 4 of them. Blastomere 1, euploid. Blastomere 2, euploid. 3, euploid. Look at blastomere 4. Now, we don't know, maybe blastomere 5 had the reciprocal chromosome numbers, right? This, I'm certain, this blastomere would not survive, nor would the daughter cell to that one. So presumably, if all the other, let's see, this is an 8-cell, was an 8-cell, so 3 of them are euploid. Let's say then, assuming that one of the remaining 4 is the reciprocal to this one, that would mean there's only 3 out of those 8 cells that would be euploid. Is that right? No, that's not right. 6. I can't do that. Yes. That should be okay, as long as these 3 as well as the other 3 are euploid. So let's take a break.